Okay, g'day everyone and welcome to this coffee science seminar. My name's David Jordan. I'll be the facilitator today. And Chris, you can move on to the next slide. So we'll begin with an acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Next slide. Okay, so today's seminar is scheduled from 12 to 1 p.m. After the presentation, we're going to hold a question and answer session. It's really important when you type your questions and answers that you do this through the Q&A rather than through the chat, and that'll make it easy for us to capture those and to respond to them. Okay, next slide. So I'm going to today introduce Chris Lambridis. I've known Chris for a long time now. We both were doing our PhDs at University of Queensland back in, too long ago, in the mid-90s. So we shared an office, a very small office with four of us, or three of us, I think. So it was, I know Chris quite well, and he's done amazing things over his career. And potentially the work that he's talking about today will be the thing that he's done that will have the most impact. So he has had a long career in plant breeding. After graduating from UQ, he spent almost a decade for Pioneer Australia breeding hybrid cultivars, particularly sunflowers. And in 2003, he did a PhD with UQ and CGIAR, which combined molecular breeding and physiology in mung beans. His research has led to several commercial releases, and he was recently part of a UQ team that released a solid resistant leukina. He has a passion for plant breeding and education. He's a course coordinator of the plant breeding course, one of the plant breeding courses, and since 2015 he's been a team leader of the BPAT project, which is a project funded by the Gates Foundation to evaluate plant breeding programs to improve their performance. So with that, I'll pass over to Chris and he can take over from here.
Well, thank you very much, Dave, and very nice of you to introduce me with those nice words. And as you said, we go way back. It's hard to believe it's almost 30 years since we first met in Cunningham Lab. It's also appropriate that Dave introduces this session, because if it wasn't for Dave, I wouldn't be associated with the BPAT project, a BMGF funded project. He and Graham introduced me to the tender that was on offer, and we'll talk a little bit about that later on. Before I start, I too acknowledge the traditional owners and their custodianship of the lands of which we meet today, and pay my respect to their ancestors and their descendants. So, as a regular part of our BPAT project, we do interviews across the world, and just recently I did a welcome to country, acknowledgement to country, to an African program in Mozambique. And we received some very glowing comments, feedback after I gave that acknowledgement. And so well done, UQ. The particular breeder said they were particularly touched by that particular part of the BPAT process. Okay, breeding program assessment tool. Sounds a bit dry. I'm hoping we can pep it up and give you an interesting talk today. Okay, so just by way of background, effectively this tool came together to make an assessment of CGIAR and NARS program. So I think it's worth me just giving you a quick definition of what CGIAR is and NARS is, so that you can have a bit of an understanding, because if you're a plant breeder, you understand these terms very well. And if you're not, and if you don't work internationally, they'll be a bit of a mystery to you. So the CGIAR, Consultative Group for International Agricultural Research, was actually formed in about 1971. And it was when a series of donors got together with the idea of improving agricultural productivity to alleviate poverty in the developing world. And so there I've got some donors listed, mostly developed countries. USA is clearly the biggest donor. BMGF is one of the biggest American donors, but you'll see there that Australia is listed there. ACIAR is the organisation that makes most of the donations. So part of the process of forming the CGIAR, several institutions were formed, and I'll give you a list of those institutions very shortly. Their goal was to make large investments into breeding programs of staple food crops. So you can see how the strategy of trying to alleviate poverty goes hand in hand with developing functioning plant breeding programs. And the CG, I'll abbreviate it CG. In fact, it's got a new name. It's called the 1CG. They have a clear mandate to help support the NARS breeding programs. Okay, so what are NARS? NARS are the government institutions that operate within countries in the developing world. So, for example, Dave and Emma have a Mercy project, and they're working with the NARS there in Ethiopia. EIAR, the Ethiopian Institute of Agricultural Research. So NARS is this generic term that gets referred to any in-country organisation that is involved with agricultural productivity. Okay, so we're now going to call the CG 1CG. And so I mentioned institutions that were formed in the CG system, and here are some of the institutions that are involved in plant breeding. And their start dates and the crops that they breed. And you'll see that they're mostly staple food crops, with the exception here of Brachiaria, which is, of course, is not a staple food crop, but it does feed animals. And of course, animals are very important for human nutrition. So CIMMIT is the oldest CG centre, formed in 1943, and ACIAR the most recent. I've regrettably had to put ICRASAT in red there, because with the new overhaul of the CG system, ICRASAT was one institution that decided not to join 1CG. Okay, so breeding program assessment tool, how did it evolve? The project is a BMGF funded program, and it began about 10 years ago when the BMGF Ag Development Team, so BMGF, Bill and Melinda Gates Foundation, Ag Development Team, wanted to benchmark the CG breeding programs. They were making quite a few investments, but they just didn't know where they should put their money. So they thought, why don't we benchmark the programs and develop a tool for assessing the status of those breeding programs? So so they then decided to contract McKinsey, a large consultancy firm, to build a tool. So then McKinsey contracted 2 commercial plant breeders, Bill Angus and Mark Walton, and hence the BPAT tool was made. In 2015, they put out a tender. Dave and Graham made me aware of the tender, so we put together a project. Dave helped me put together the very first BPAT project, and we were successful in 2015 in getting the first BPAT grant. So phase 1 has run from 2015 to 2020, and fortunately we were able to get a second phase that started last year. BMGF have a series of investments that they make to improve plant breeding programs in the CG system. And the BPAT project works very closely with the Excellence in Breeding platform. And I'll explain a little bit more about that later on. Okay, so what is BPAT? It's a series of survey tools used to evaluate the effectiveness of plant breeding programs. But what BMGF wanted, they said to themselves, right, where do we see plant breeding programs done very well? And they looked at the private sector model. And that's what they wanted to put together. An evaluation tool that used the principles that apply in the private sector. So they wanted to identify strengths and areas for improvement, but then use these criteria commonly used to evaluate commercial plant breeding programs. Clear target product profiles. So any good plant breeding program has to know exactly what it wants to do. A clear target product profile. They need to have a clear understanding of consumers, customers, and the marketplace, knowing what the product requirements are. Of course, they're always driving better return on investment, so they have to have these breeding programs operate effectively and efficiently, and cost effectively, most importantly. And there's always a focus of continual improvement. Okay, so when we took over the BPAT tool, we came up with this little motto, maximizing genetic gain, and you'll see that throughout this presentation. Okay, what form did we receive the BPAT tool in? In this is effectively what we got. We got a series of single worksheets out of an Excel file with 100 questions on them. And the first thing we said, we just can't possibly operate like this. So you can imagine spending 2 days with a breeding program, wading through a form like this, trying to work out where you are and where you had to go next. So at the beginning of UQ's handling of the BPAT tool, we had Sandra McCullough was employed, our data manager, and her very first job was to convert the tool into something that was very usable. Okay, so this is what we have today. We have a series of tools that have been made using Access software, and they're very, very easy to use. We had all sorts of problems trying to build the tools in Excel. They weren't particularly stable. So we moved to Access, and we've really had a few problems since. So you can see that here we have our motto in each of the survey tools. So this is a pre-visit institution questionnaire form that we have. This is the pre-visit breeding program questionnaire. And then this is the tool that the assessors have to assess the breeding programs. So by now you've realised that what this is all about is going in and making a thorough investigation of a plant breeding program to try and bring them up to a high level of functionality and effectively modernise their operation. Okay, so just to give you an overview of the process, we have about a 10-week process. We introduce ourselves to the breeders. We have an introductory meeting. We send out the pre-visit surveys. This is an opportunity for the assessment team to gather a lot of information without actually having any one-on-one interviews. Then, after about a month, we conduct the interviews and we make our assessments. We then, soon after we make the assessments, we give a short PowerPoint feedback presentation, and then we take about a month to write a detailed report, and then we hand over our scorecards of the assessment. So this slide is just to demonstrate that in the original phase 1 of the grant, what we used to do is set up these meetings and get our data collected, and then we would actually fly to the plant breeding program. So most of the phase 1 assessments were done where we took the assessment teams from around the world and we flew into the institution to conduct the assessments. Of course, the pandemic has prevented us from doing that, so we've now have the new norm where we do everything virtually. Okay, I'll just take a quick mouthful. To make this presentation as widely applicable as I could to the listeners, I wanted to leave out as much of the technical detail as I could. So what I've done here is I put in words what effectively is the breeder's equation, which all breeding programs should adhere to. So let me just read this out to you. To achieve genetic gain per unit of time, a breeder needs to have a clear product profile, make statistically rigorous decisions based on high quality data sets to select the best genotypes from a large population, enabling the selection of plants with superior traits. And then the process is repeated. So I hope you can see my cursor. So basically, cleared product profiles, you select the appropriate germplasm of the appropriate genetic diversity that you need. You intercross the material, you create new recombinants or new plants. You select the best and you start the whole process all over again. And so what you're trying to do in a plant breeding program is go through this cycle as quickly as you possibly can. And of course, down here, this is where you siphon off good materials to identify products that can be released to farmers. So effectively what I've just tried to do there is give you a quick plant breeding lesson. And it's based on what breeders will know well as the breeder's equation. Okay, so I'm going to go through the main survey tool that the assessors use. It effectively consists of 150 questions divided into 11 components. I'm going to go through these components very quickly. We start off by saying, okay, what is the breeding program? What are the objectives and what are their product profiles? So here we understand, well, how well have they defined the target? The most important thing of a breeding program, you've got to define your target. You have to know exactly what you're breeding for. Then we say, okay, given that target, what breeding team have you assembled? What disciplinary support have you put together to address these breeding objectives? Okay, and then we say, well, given those two things, what infrastructure do you have in place? Do you have suitable facilities to achieve the goals of your breeding program? Then we move into the nitty-gritty of breeding methodology. We ask a whole series of questions about the breeder's equation. Now you're all breeders now because I've explained what the breeder's equation is all about. And here it is represented again down here. Basically, we're interrogating all aspects of this process. And what we're particularly interested in looking for is statistical rigor. Plant breeding is all about statistical rigor. Then we move on to, just move my face off the, oh, it's not going to move for me. There we go. Okay. Then we move to a series of questions on product development and release. So what are the processes involved in actually using the data sets that have been derived in the breeding methodology to release material? What breeder seed production processes are in place to develop good quality seeds for farmers to use? Okay, so I'll go to the next set of components. Components 6 to 11. Then we assess, okay, given that this program has a certain budget and it's been operating for a certain number of years, what has been the impact of the program, which is really quite important. Then we ask about innovation, because, you know, the whole purpose of doing these evaluations is that we want to modernize these CG and NARS plant breeding programs. We want them to be operating as effectively as they possibly can so that they can be developing products for farmers so that they can eventually get greater productivity, greater profitability, and hopefully contribute to alleviation of poverty. So we want to know how innovative have they been? Have they been modernising their breeding programs? One of the most important things about the whole process is the CG have this very difficult plant breeding task, but ultimately they don't release the varieties. They have to hand their materials over to the national governments, to the NARS programs. So this is the part of the system that is a little precarious and needs a lot of attention. So what we go in and do, we try and understand how good is that network? How well is it functioning? Then we ask questions about the alignment. How well is this breeding program aligned to other like programs? Do they collaborate? Are they learning from others? Do they link in with international programs? Then we try and understand budget and finance. Are they using their resources appropriately? Do they cost their operations? Are they using the UQ costing tool? The last component is a whole series of questions on organisational effectiveness. And we have a specific company that we work with, Morgan Clarkson, led by Vanda Morgan. She's an organisational effectiveness expert. She conducts that part of the assessment, because you can be the best breeding program in the world, but if you don't have good support from your organisation, you just can't function properly. Okay, so let's have a look at some of the features of our assessment form. Okay, so that's what it looks like. Everything's broken up here into the components. It's all very user-friendly. Easy data entry interface. These are all access databases. Each question that we ask, the assessors give a score of 1 to 4, 1 being poor, up to 4 being very good. So within the form, the assessor can click on this tab here and they can come up with the criteria that underpin the question. So that way it makes it easy for the assessor to give a score. Now, because we ask for pre-visit survey data, so that means weeks before we've actually conducted the assessment, we've already got a good idea of what the breeding program looks like. But now this form, by clicking on this tab, we can bring up right in front of the question, what did the breeders, how did the breeders respond to that particular question? Because we've asked related questions in the pre-visit survey that will allow the assessor then to listen to the answer live one-on-one, but also incorporate the information that the breeding team gave before the assessment began. There's even a spell checker in it. Very important for some assessors. Okay, and then an ability to change, add underlying data, crops, locations, institutions, etc. Okay, and we can create subsets of questions. So the form is about 150, but if we go to a really small program where we think perhaps 10, 15, 20% of the questions might not be applicable, we can actually subset the form and just use a reduced subset of the questions. Okay, so quite often we'll have 3 or 4 assessors assessing a program. So we have the capability that at the end of the assessment, given that each assessor fills out their own form, okay, independently at the time, okay, they each fill out their own form. The forms can be merged together. So here's a demonstration of that process. Again, this is all embedded in the one access database, okay? So this was a recent BPAT that we did. And so here, this is the files are being loaded. when we were doing live interviews, at the end of the day, we would sit around a table and we would moderate scores. But that's less difficult. That's more difficult to do now because we have assessors all over the world and operating at very odd times of the day. So Sandra developed this really nice feature where you can display all the scores and we can see if there's any moderation needed. In this particular set of questions, you can see that most of the scores are pretty similar between the assessors. And so therefore, we don't have to, there's probably no need to moderate. Occasionally we get the disparity in the scores is such that we really have to review them. And at least one assessor does decide to make a change, although they don't have to. If they feel that their score is more of their thinking, then that's quite okay. There's no obligation to change a score. Okay, so we had 4 assessor score sheets. What the database can then do for you is we can then combine all the scores into a single report. So that way the breeders receive everybody's comment and score, and they'll get an average score of the 4 assessors in this case, but they'll see everybody's comments. Okay, I said to you before that we work hand in hand with another BMGF investment, the Excellence in Breeding platform. Our job is the relatively easy one. We go in, we review a breeding program, and we make recommendations for improvement. The Excellence in Breeding platform are now set up to offer expertise to help the breeding institutions write program improvement plans and start to address some of the BPAT concerns that were raised. So again, with this, all this is within the same form, we can then subset the scores. So in this case, we're going to prepare an EIB report where the scores were less than 2.5. Again, now remember, scores of 3 and 4 are very good. So basically what we're saying is, let's find out where the problem areas were. Let's assign a priority. Let's assign a module leader. So the EIB has 5 modules. So we assign a module leader to the particular issue. In this case, it was EIB module 2. And so this report gets sent straight to the module leader. And so they know exactly where they need to start providing assistance. Okay, so some outputs of our assessments. A detailed report outlining program strengths and areas for improvement. So that's a written document. We give the breeders assessor scorecards, all combined in a single report as a PDF. We give them the average scores of each of the assessors. We give the EIB a report, and this whole process initiates the process of writing a program improvement plan. And quite often the BPAT team members are involved in writing the program improvement plan. Okay, so what have we done so far? Since we started our very first BPAT assessment in 2016, we have now assessed 84 plant breeding programs. Effectively, that represents 84 assessments, 65 breeding programs, because about 30% of them we've actually done twice. Okay, and from March 2020, all the assessments were done virtually, and that's indicated by the mustard color here. So these were all live assessments. And then, of course, due to the pandemic, we've had to revert to virtual assessments. You can see that the target locations, most of them in Africa, 20% in Asia, and some in Latin America. All the important staple food crops have been reviewed. Okay, so let's go through some of the things that we found, some of the strengths of the CGIAR programs. Without exception, always identified dedicated, experienced breeding teams. and for that matter, the disciplinary support to the teams, always, really, really good quality. They all have a very good understanding of production constraints confronting farmers, smallholder farmers. Often they publish high quality science, and publication is a bit of a priority for quite a few of them. They have very good access to germplasm, gene banks. So the CG has done a really good job of creating these gene banks and making the germplasm available for the CG breeders and subsequently the NARS breeders to get the required germplasm. There have been many varietal releases, and some varieties have been highly successful, so the so-called mega varieties. But quite often they turn out to be very old varieties that were released many years ago, and just they're finding them very hard to replace. Often these programs have very strong molecular teams, but they're not always well aligned to the breeding program. The CG programs are always very well aligned to international institutions. There's always willing collaborators. Okay, I just wanted to talk about a couple of the challenges before we talk about some of the areas for improvement. I think a lot of us who are associated with these assessments talk about how difficult it is for CG breeders to do their job. Because whilst they might be based in a CG institution, let's say in Africa, for example, IITA, which is in Ibadan, Nigeria, their mandate is wide. It's not only Nigeria, they have all of West Africa, they have parts of East Africa, they have parts of Southern Africa. So trying to develop materials that have wide adaptability is a huge challenge. And sure, they do have regional outposts, smaller breeding programs, but generally they're poorly resourced. So that's a huge challenge for the CG programs. But here's another enormous problem. This particular slide demonstrates the yield gap. So this slide it's from using FAO data that Tiago Mendes showed us last week when we assessed the SIP potato breeding program, and it demonstrates the yield gap. So this line here tracks the productivity of potato in the Netherlands, but here this data tracks the productivity of potato in Africa, and so there is this enormous yield gap. One of the principal reasons is they have lots of disease and insect pressure, pest and disease pressure, but also, probably most importantly, many, if not most, farmers in Africa do not use fertilizer other than organic manures. So they do not use fertilizer. So how do you breed products that are going to have to respond to a climate change, a changing climate, minimal inputs of fertilizer with enormous disease pressure? Here's another thing that I wanted to make you aware of. Some of these crops are incredibly difficult to breed. So this is an example of the Matoki banana breeding program in East Africa run by IITA and NARO. So they have really complex genetics that they have to deal with. So as you all know, banana is sterile. It has to be sterile. When you eat the fruit, you can't be chomping into seeds. So the starting material in their breeding program is a triploid sterile plant. And these are typically land races. So then they try and cross it to a diploid source of, say, disease resistance. Now, from that primary cross, they have some of the progeny are diploid, some are triploid, and some are tetraploid. So what they do is they take this tetraploid material only to bring it down for the secondary cross, where they cross it to an improved diploid to finally come up with a sterile triploid. Now, that is such a complicated breeding strategy, but even more alarming is that they effectively use the same parents at each of these steps, and they just create new recombinants every year. So really, no recycling. To remember, I had that cycle that's very important in plant breeding programs, where you're gathering favorable alleles each generation. You're bringing together the genes that you want, and every year you're adding new genes to add to greater productivity. Okay, so that's an enormous problem. But here's the thing. The progenitors of banana are actually diploid bananas, and they reside at our doorstep in Southeast Asia, primarily in Indonesia. So there is an opportunity for UQ, and I had the pleasure of attending a meeting held by Nina recently about the opportunity for Australia to get involved in banana breeding, because in Australia, 95% of commercial cultivation is sown to a single cultivar, Cavendish, 95%. How vulnerable is the industry? The question I ask. So an opportunity for UQ to contribute. Another opportunity for UQ to contribute. Invariably, most of these staple food crops have problems with quality. Even if a farmer can get more yield through having better disease resistance, if the quality is not right, the farmer will not grow that crop, that variety. So we see this almost unilaterally across all the programs, is that quite often the varieties fail to meet quality attributes. So what is needed is high throughput screening assays to screen for quality attributes. Now and I'm not a quality scientist, but I know that's complicated. You've got aroma, you've got flavor, you've got mouthfeel, you've got a whole lot of nutritional aspects. But what tends to happen, because the breeders don't have high throughput systems, what they do is they'll go through their breeding program, and once they've got 15, 20, 30 clones, they'll then start to screen for quality. But then they may have thrown away all the good quality attributes way before they got to the end of the pipeline. So I think an opportunity for UQ with all our expertise in breeding and food science. So what is really needed is high throughput assays for determining quality. Okay, so we're going to move to areas for improvement that we identify, or issues in, I'm going to make some generalizations about the breeding programs. So we were confronted in many cases with what we call institutional inertia. The breeders just doing what they've been doing for 30, 40 years, and procedures being handed down to them. Poorly defined product profiles. Often the breeders would breed for too many profiles and too many traits, stretching their resources to the limit. So remember this breeding process that I explained to you based on the breeder's equation. You really have to have a very clear target, and not too many of them. If you do have too many of them, you just can't be effective. And unfortunately, you can't please all of the people all of the time. A clear lack of a quantitative plant breeding approach. They lack the statistical rigor that I mentioned that plant breeding programs need. Unable to really adequately define the target population of environments, because they're in country, but their targets are outside of the country that they're operating in. And so the CGIAR network is not strong enough for them to interrogate this properly. Quite often, when CG programs send trials out for testing in the NARS research programs, they don't get good quality data back. That's a huge limitation. Data analysis, unsophisticated data analysis. So again, not providing the statistical rigor needed. Quite often, research stations had suboptimal field nurseries. Often we would hear breeding programs say, right, we need to breed for drought tolerance, but there'd be no physiology expertise to inform the breeding program. Seed systems, a huge issue. So there have been many investments by the donors to improve seed systems. That is, once a variety is developed, getting the seeds, the necessary seeds to farmers. Quite often too many cultivars are released. A big problem, poor monitoring of post-release performance. But you can imagine if you're, let's say you've released a new cassava variety in Nigeria where there are 5 million hectares, that means 5 million farmers because they're small holders, about a hectare each. If varieties get planted amongst those 5 million farmers, how do you track that? It's a very difficult process. We talk about the breeder's burden, a focus on publications rather than product development. Many of them have had a funding crisis. The funding model in the CG has been topsy-turvy for many years. And the realization that we now have of one CG is aimed to address that. Poor data return from NARS, I think I've already mentioned that. Testing done at just one location, the institutional headquarters. Organisational issues, poor succession planning, high turnover of staff, lack of mentorship in plant breeding. Okay, we've done, as we said, 30% of our assessments are reassessments. And can we demonstrate improvement? Yes, absolutely we can. A lot of effort, largely due to the efforts of the EIB in the definition of product profiles. Still some work to do, but a big improvement. There's been many examples of a quantitative plant breeding approach. So that means that they're starting to get the statistical rigor that they need. They're starting to plant more trials, not at a single location, multi-environment trials to get good, accurate data. Starting to mechanize electronic data capture. Starting to actually mechanize some of their planting and harvesting operations, which are largely not done. And quite a few of the institutions, at the recommendation of the BPAT process, have appointed global breeding leads to offer mentorship in plant breeding. Okay, so what's next? We're going to continue assessing CG/NARS programs, particularly the networks. And we're going to have a real focus on building online educational modules for the developing world, really targeting those NARS programs where the opportunities to do plant breeding, to have plant breeding education may be limited. And it's a pleasure to introduce our brand new staff member, Zulfi Chahufa, who has just come from Ag Research in New Zealand to join the BPAT team. And we hope to put together an online plant breeding textbook. The textbooks that we learned from in our degrees didn't really prepare us well for the real world. Okay, we're winding up now. The BPAT assessment is only as good as the team of assessors, and I am very fortunate. I'm the facilitator of the process, but I get to invite the best breeding experts that are available. And it's been a great pleasure to work with all of these people. I'd like to especially thank Yilma Kabedi, Mark Cooper, and Dave Jordan, who have spent additional time helping build the tools and modify the tools. Andre Drenth has been a great help too the last year or so, particularly with the RTB, so-called RTB crops, roots, tubers, banana. But also I've got to make mention of Mark Dieters here. So Mark is an ex-UQ academic and breeder. So Mark has just joined the Excellence in Breeding, but he maintains an office here at UQ. And now we've got a really good conduit with the EIB. So Mark attends most of our BPAT assessments, and now we're really getting a good flow of information to the EIB teams. And of course, the BPAT team is only as good as the support staff. I'm deeply indebted to Sandra McCullough and Juliana Thompson for making all of this happen. I absolutely couldn't run this program without them. And just to acknowledge BMGF, their contribution to these projects. So not only do they fund BPAT and EIB, they fund Dave and Emma's Mercy, John Thomas, Banana Boris work, Liz Aiken working on also banana diseases. So there's the big High Gain project with Anna Coltonau and Ian and Dave and Emma and others. So and I'm sure there are other projects that I probably missed, but incredible contribution of BMGF to this area of agricultural research. Okay, that's we're winding up some of the live assessments that we did over the years. Of course, that's now a thing of the past, and this is the way of the future. Virtual assessments. I think largely we've been successful to achieve that. And I think, Dave, I'm probably done and happy to answer questions. I didn't think I was going to speak this long anyway.
You've cut into your question time, Chris, which is unfortunate in a way because there's some really good ones. So there are some technical ones I think that would probably be good if you catch up with John Dixon about, but I thought I'd ask the most controversial one first.
Yes.
So don't you think that the CG and BMGF should immediately stop all investment in crop improvement and focus on lifting farmers' agronomic skills, reducing poverty and market access?
That's an entirely appropriate question given the yield gap that I presented. So here's the thing, that there has been a lot of genetic gain made in crops like wheat and maize, disease resistance, quite substantial benefits from breeding for disease resistance in those particular programs. And so that what is needed is a combination of effort. Okay, so there's lots of things that can happen with improving genetics, but undoubtedly it has to be in alignment with other agronomic processes, agronomic inputs. And probably that was a question from somebody like Peter Carberry, I'm guessing, but I might be wrong.
Not sure. It's an anonymous attendee.
Hey, Peter, if you're out there, I hope you're listening.
And it wasn't me. So
okay, let's move on.
Okay,
good question,
so are the this is one from Ian Godwin. So how do international programs actually deal with these issues effectively? I think that's probably, I might paraphrase, it mightn't be the question that Ian was asking, but do you see good change in response to your evaluations when you've gone back to look at people again?
Yeah, absolutely we do. I mean, it's taken time. So, let's not forget the 30, 40 years of institutional inertia. So basically, trying to get people to change their ways. It's not easy. And I think in a relatively short period of time, there's been amazing progress made. And let's not forget, our job was to make the assessments and hand over to the EIB. The EIB is really in its fledgling years. It's effectively started a year or two after the BPAT project got underway, and it's been building over the last few years, and it's really gearing up. I mean, the number of new staff is really quite amazing. So I think there is clear examples, as I demonstrated, of improvement, and it's only going to get better. And can I say that the breeders are on board? Absolutely, they're on board and they get it.
Mm-hmm. Yeah, and probably just from our experience in Ethiopia, organisational change is not easy. It's the hardest thing to do. So
yeah, good point.
There's a lot of that happening. Question from Craig: has the UQ team made any changes to the content that you were handed, and why? And I think you've touched on that, but maybe a brief answer.
Yeah, absolutely, Craig. So quite a few modifications as we really wanted to embed, the breeders equation in there, and embellished that section of the assessment. And of course, we've recently been contracted by other organisations to do assessments in Africa. So, for example, World Coffee Research. So we made a tool, a separate tool, contracted outside of the BMGF grant to assess coffee breeding in East Africa, to Ethiopia, a very important crop for them, and in Uganda.
Okay, this anonymous attendee's got another good question. So do you see a trade-off in the CG in having such a significant focus on publishing science rather than being an agent for positive change?
Yeah, okay, so the trade-off of publishing versus product development. That's been an issue, a rather contentious issue for a lot of the scientists. I think it just needs to be institutionally put to the teams that there should be a priority. And I think if you arrange the skills in the team appropriately, you can achieve both. You can actually have good product development teams and also write good papers. Right, Dave? That's what you do.
Try. Okay, another one from Michael Advardi, and it sort of probably captures a number of fields besides the one he's mentioning, but poor integration of molecular breeding with field activities and the goals is a common problem in developing countries. Can you, what do you advise in terms of better alignment?
Yeah, so that's a good point, Michael. Basically, everybody has to come on board together as a team, and we came up with a term for that. It was product-driven disciplinary support. In other words, everybody comes together to develop the product. But quite often what would happen is that they would remain in silos. And so that just gets down to plant breeding leadership within the institution. And that's why we spoke of mentorship. And very good to see that there are now global breeding leads at a lot of the institutions that weren't in place before. So it's just about leadership, I think, Michael.
I'd probably add there, it's about accurately measuring the return on investment as well. So I think often markers are used, or molecular tools are used when the return doesn't justify the investment, and vice versa, where they should be used. Okay, let me have a look. Ha ha! No, I won't go into that one. Right. It's difficult for breeders in this will probably be the last question. It's difficult for breeders in Australia to breed for wide adaptation. What works in WA is not necessarily suitable in Queensland. I think this is, I agree with Ian's comment here or question here. This is a real issue for the CG system, trying to breed across such large environments. Do you want to make a comment?
Yeah, for sure. So that's where a lot of the EIB and the BPAT focus in coming years is to develop the CG NARS networks. We have to engage the NARS programs to participate, and that's where you'll get value. The problem is, if you go to countries like Ethiopia and Uganda, they have very strong sponsorship of plant breeding activities. If you go to a country like Tanzania, they don't. So it really requires, where there's a will, there's a way. So the NARS really have to demonstrate that they want this and they need this. So this is the challenge, is to build these CG NARS networks. And I think there's a lot of effort going into that, particularly within the EIB.
Okay, so I think that brings us to the end of our questions there. We don't do applause on Zoom, I guess, but I think everyone, but judging from the questions, there's a lot of interest in this topic, and I'm sure Chris will get back to you on those particular questions. And I'd like to thank him for giving us such a great seminar. And if we could move to the next slide, Chris. Keep going. We've done that. Okay, so just to advertise our next science seminar, one of Coffee's stars, Ben Hayes, is going to be talking about breeding Australian cattle for production in the year 2050. So put that in your calendars, everyone. Next slide, please, Chris. So you can scan this QR code to go to the Coffee Science seminar page and catch up and stuff there. And also you can scan to give us feedback on our seminars and how we might change things. So please do that if you feel that there's opportunities for change. So with that, I will say goodbye to everyone, I think. And Chris, if you want to say a few words, you're welcome to thank everyone.
Yeah, well, certainly. Thanks for those excellent questions, and happy to. Oh, one thing I didn't actually get around to saying is that all our tools are freely available, so you can modify them for whatever purpose you want. So just contact us, and the tools are available to anybody who thinks that they might want them. And thank you, Dave, in particular, for being a very, very good host and saying nice things about me.
Oh, it's easy to do, Chris. I don't think I know of anyone who's got a bad word to say about you, Chris.
Oh, don't know about that. Okay.
Okay. Thank you, everyone.
Thanks.